towards or away from the heart. 7 do veins carry blood towards or away from the heart? 8 describe how blood is pushed out of the heart. 9 which structure keeps oxygenated blood and deoxygenated blood separate from one another in the heart? Suggest why it is important for them to be kept separate. 10 if you have a diagram of a vertical section of the heart in your notebook, add labels to it to show which of the one-way valves are atrioventricular valves, and which are semilunar valves. 11 explain, in your own words, why the walls of the atria are thinner than the walls of the ventricles. Coronary arteries. Figure 9.5 shows the external structure of a human heart. You can see that there are blood vessels on the outside of the heart. They are called the coronary arteries. These vessels supply blood to the heart muscles. Figure 9.5, external appearance of a human heart. It may seem odd that this is necessary when the heart is full of blood. However, the muscle of the heart is so thick that the nutrients and oxygen in the blood inside the heart would not be able to diffuse to all the muscle quickly enough. The heart muscle needs a constant supply of nutrients and oxygen so that it can undergo aerobic respiration. This releases energy that the muscle uses to contract. The coronary arteries supply the nutrients and oxygen that the muscle needs. Coronary arteries can get blocked. In some people, cholesterol deposits build up inside the walls of arteries, including the coronary arteries. These deposits make the artery wall stiffer and the lumen narrower, so it is more difficult for blood to flow through. Blood clots can form. If the coronary artery is blocked, the cardiac muscle does not get any oxygen. The muscle cells cannot respire aerobically, so they cannot obtain energy to allow them to contract. The heart therefore stops beating. Figure 9.6. How coronary heart disease is caused. Blockage of the coronary arteries is called coronary heart disease, or CHD for short. It is a very common cause of illness and death, especially in developed countries. We know several factors that increase a person's risk of getting coronary heart disease. Preventing CHD. CHD is one of the commonest causes of death in many countries. No one can completely eliminate the risk of developing CHD, but there's a lot that can be done to reduce this risk. The most obvious thing you can do is not to smoke cigarettes. Smoking greatly increases the chances of developing CHD, as well as many other unpleasant and dangerous health problems. Taking care over your diet is also a good thing to do. A diet that is high in saturated fats is linked with an increase in the concentration of cholesterol in a person's blood, and this in turn increases the risk of CHD. Meat is higher in saturated fats than plant-based foods, so one simple way to reduce the risk of CHD is to substitute animal fats with plant oils when you are cooking. Fast foods are often high in animal fat, so these should be eaten in moderation. Regular exercise has a very beneficial effect on many parts of the body, including the heart. Most people can find some kind of exercise that they enjoy. Exercise helps to keep you fit, prevents excessive weight gain and decreases blood pressure. It also has a feel-good effect by helping to clear your mind of things that may be worrying you and causing the release of chemicals in the brain that increase feelings of well-being. Figure 9.7. Regular exercise can decrease the risk of developing CHD. Heartbeat. You may be able to feel your heart beating if you put your hand on your chest. Most people's hearts beat about 60 to 75 times a minute when they are resting. If you put your head against a friend's chest or use a stethoscope, you can also hear the sounds of the valves closing with each heartbeat. They sound rather like lub-dub. Each complete lub-dub represents one heartbeat. A good way to measure the rate of your heartbeat is to take your pulse rate. A pulse is the expansion and relaxation of an artery, caused by the heart pushing blood through it. Your pulse rate is therefore the same as your heart rate. You can find a pulse wherever there is an artery fairly near to the surface of the skin. Two good places are inside your wrist and just to the side of the big tendons in your neck. There are also phone apps and smart watches that can automatically record your pulse rate. In a hospital, the activity of the heart can be recorded as an ECG. This stands for electrocardiograph. Little electrodes are stuck onto the person's body, and the electrical activity in the heart is recorded. The activity is recorded as a graph. An example of a normal ECG is shown in Figure 9.9. Figure 9.8. The electrodes fixed to the man's body will record the activity of his heart. Figure 9.9. A normal ECG. You can think of it as a graph with time on the x-axis and electrical activity on the y-axis. The points labeled P, Q, R, S and T represent different stages in one heartbeat. How the heart beats. The heart beats as the muscles in its walls contract and relax. When they contract, the heart becomes smaller, squeezing blood out. When they relax, the heart becomes larger, allowing blood to flow into the atria and ventricles. Figure 9.10. How the heart beats. We have seen that there is an atrioventricular valve between the left atrium and the left ventricle, and another between the right atrium and ventricle. The valve on the left-hand side of the heart is made of two parts, and is called the bicuspid valve, or the mitral valve. The valve on the right-hand side has three parts, and is called the tricuspid valve. As the walls of the atria contract, they increase the pressure of the blood in the atria. This pushes down on the atrioventricular valves, swinging them open like doors and allowing blood to flow through, down into the ventricles. But when the ventricles contract, the valves are pushed closed again by the pressure of the blood in the ventricles. The tendons attached to them stop them from swinging up too far, they have to stop in the closed position. Questions. 12. Describe where you would find coronary arteries and explain their function. 13. Explain why coronary arteries can sometimes become blocked. 14. List 3 different ways you can measure your heart rate. 15. Look at the ECG in Figure 9.9. A. Use the scale to find the mean time between two Q spikes. Use your answer to A to calculate the person's heart rate in beats per minute. 16. Explain how the septum and the bicuspid valve help to ensure that fully oxygenated blood leaves the heart in the aorta at high pressure. Experimental Skills 9.1. Investigating the effect of physical activity on heart rate. In this investigation, you will measure your pulse rate before and after exercise. You will practice measuring carefully, recording results clearly and using them to draw a line graph. You will need a timer or a smart watch that can measure pulse rate. Safety. If you are worried that doing exercise might make you ill, do not do it. You can measure someone else's pulse rate instead. Before you start, practice measuring pulse rate. Try placing 2 fingers on your own neck, rest them gently closely to one of the big tendons, and try to feel the beating of your pulse. When you are confident that you can feel the pulse, try to find it on your partner's neck. If you are going to use a smart watch to measure pulse rate, make sure that you know how to use it. Figure 9.11. Feeling the pulse in the neck. Now practice counting how many pulses there are in 30 seconds. You may find it helpful to have a partner to call out start counting and stop counting. You can multiply the number of pulses you have counted by 2 to find the pulse rate in beats per minute. Method. Work with a partner. 1. Read through the method and construct a table to fill in your results. 2. Ask your partner to sit very still and relax. After a few minutes, measure your partner's pulse rate. Do this 3 times. Record the 3 values. 3. Ask your partner to do some exercise, such as raising their arms over their head vigorously for 1 minute, or running on the spot. When they stop, measure and record their pulse rate again. Do this every 2 minutes, until their pulse rate goes back to the resting value. Questions. 1. Use your results to draw a line graph. Put time on the x-axis and pulse rate on the y-axis. Mark on the x-axis where exercise began and ended. Use a ruler to join the points with straight lines. 2. Use your table or graph to describe, in words, how exercise affected your partner's pulse rate. 3. Explain why the pulse rate increases when you exercise. Think about what your muscles do when you exercise. How the muscles get the energy to do this. How the muscles are supplied with their needs. 9.3 Blood vessels. There are 3 main kinds of blood vessel: arteries, capillaries and veins. Arteries carry blood away from the heart. They divide again and eventually form very tiny vessels called capillaries. Capillaries deliver blood containing oxygen and nutrients close to every cell in the body. The capillaries gradually join up with one another to form large vessels called veins. Veins carry blood towards the heart. They have valves to help to keep the blood flowing in the correct direction. Figure 9.12. Sections through an artery, capillary and vein. The drawings are not to scale in reality. Capillaries are much smaller than either arteries or veins. Figure 9.13. Arteries divide to form capillaries, which join up again to form veins. Arteries. When blood flows out of the heart, it enters the arteries. This blood is at high pressure, because it has just been forced out of the heart by the contraction of the muscles of the ventricles. Arteries need very strong walls to withstand the high pressure of the blood flowing through them. The blood does not flow smoothly through the arteries. It pulses through. The pressure of the blood is high when the muscles in the ventricles contract, and reduces as these muscles relax. The arteries have elastic tissue in their walls which can stretch and recoil with the force of the blood. This helps to make the flow of blood smoother. You can feel your arteries stretch and recoil when you feel your pulse in your neck or wrist. Capillaries. The arteries gradually divide to form smaller and smaller vessels. These are the capillaries. The capillaries are very small and penetrate to every part of the body. No cell is very far away from a capillary. Figure 9.14. A capillary, shown in blue, makes its way through muscle tissue. The function of the capillaries is to take nutrients, oxygen and other materials to all the cells in the body, and to take away their waste materials. To do this, their walls must be very thin so that substances can get in and out of the capillaries easily. The walls of the smallest capillaries are only 1 cell thick. Veins. The capillaries gradually join up again to form veins. By the time the blood gets to the veins, it is at a much lower pressure than it was in the arteries. The blood flows more slowly and smoothly than it did in the arteries. There is no need for veins to have such thick, strong, elastic walls. If the veins were narrow, this would slow down the blood even more. To help keep the blood moving easily through them, the space inside the veins, the lumen, is much wider than the lumen of the arteries. Veins have valves in them to stop the blood flowing backwards. Valves are not needed in the arteries, because the pressure produced by the heart keeps blood moving forwards through them. Figure 9.15. Valves in a vein. The veins are like pockets set into the wall of the vein. Blood is also kept moving in the veins by the contraction of muscles around them. The large veins in your legs are squeezed by your leg muscles when you walk. This helps to push the blood back up to your heart. Table 9.1 summarizes the structure and function of arteries, capillaries and veins. Blood vessel. Function. Structure of wall. Width of lumen, how structure fits function. Arteries. Carry blood away from the heart. Thick and strong, containing muscle and elastic tissue. Relatively narrow, it varies with heartbeat, because the walls can stretch and recoil. Strength and elasticity needed to withstand the high pressure and pulsing of the blood, as it is pumped through the arteries by the heart capillary supply all cells with their requirements, and take away waste products. Very thin, only 1 cell thick. Very narrow, just wide enough for a red blood cell to pass through. No need for strong walls, as most of the blood pressure has been lost. Thin walls and narrow lumen bring blood into close contact with body tissues, veins. Return blood to the heart quite thin, containing far less muscle and elastic tissue than arteries. Wide, contains valves. No need for strong walls, as most of the blood pressure has been lost. Wide lumen offers less resistance to blood flow, valves prevent backflow. Table 9.1, Structure and functions of arteries, capillaries and veins. Naming blood vessels. Figures 9.16 illustrates the positions of the main arteries and veins in the body. Figure 9.16, Plan of the main blood vessels in the human body. Red represents oxygenated blood, and purple represents deoxygenated blood. Each organ of the body, except the lungs, is supplied with oxygenated blood from an artery. Deoxygenated blood is taken away by a vein. The artery and vein are named according to the organ with which they are connected. For example, the blood vessels of the kidneys are the renal artery and vein. The liver has 2 blood vessels supplying it with blood. The first is the hepatic artery, which supplies oxygen. The second is the hepatic portal vein. This vein brings blood from the digestive system, so that the liver can process the food which has been absorbed, before it travels to other parts of the body. All the blood leaves the liver in the hepatic veins. Figure 9.17, The main arteries and veins of the human body. Activity 9.1. Comparing plant and animal transport systems. Work in a group of 2, 3 or 4 for this activity. In Chapter 8, you learnt about how xylem and phloem are involved in transporting substances in plants. Now, you also know how blood vessels transport substances in animals. In your group, think of at least 2 similarities between the transport system of a plant and the transport system of a human. Now think of at least 2 differences between them. Work with the other groups in your class to combine all of your ideas to produce a comparison between the transport systems in a plant and in a human. Perhaps this can go on display in your classroom. Reflection. Think about how you worked to construct your comparison. How well do you think you contributed to your group's work? Did other people in the group help you in any way? Did you help others? Do you think that working in a group like this helps you to understand or remember information better than working on your own? Questions. 17. Name the type of blood vessel that has the thickest wall. Bars. The thinnest wall contains valves. 18. Look at figure 9.16. A. Where does deoxygenated blood become oxygenated? B. Where does oxygenated blood become deoxygenated? C. Most arteries carry oxygenated blood. Name one artery that carries deoxygenated blood. 19. Explain why the walls of arteries need to contain more elastic tissue than the walls of veins. 20. Outline how the supply of blood to the liver differs from the supply of blood to other body organs. 9.4. Blood. Blood is a liquid that contains cells. The liquid part of blood is called plasma. Most of the cells that are carried in this liquid are red blood cells. A much smaller number are white blood cells. There are also small cell fragments called platelets. They are formed from special cells in the bone marrow. Figure 9.18, The components of blood. These cells and cell fragments float in a pale yellow liquid called plasma. Figure 9.19, A photomicrograph of blood. The blood has been colored with a stain to make the different kinds of cells show up clearly. Red blood cells. Red blood cells are red because they contain the pigment hemoglobin. This carries oxygen. Hemoglobin is a protein and contains iron. It is this iron that readily combines with oxygen where the oxygen concentration is high. This happens where blood capillaries take the blood close to the alveoli in the lungs. Hemoglobin that is combined with oxygen is called oxyhemoglobin. Oxyhemoglobin readily releases its oxygen where the oxygen supply is low. This happens when the blood passes through capillaries close to cells that are respiring, using up oxygen. Red blood cells are unusual because they do not possess a nucleus or mitochondria. The lack of a nucleus in a red blood cell means that there is more space for packing in millions of molecules of hemoglobin. Another unusual feature of red blood cells is their shape. They are biconcave discs like a flat disc that has been pinched in on both sides. This, together with their small size, gives them a relatively large surface area compared with their volume. This high surface area to volume ratio speeds up the rate at which oxygen can diffuse in and out of the red blood cell. The small size of the red blood cell is also useful in enabling it to squeeze through even the tiniest capillaries. This means that oxygen can be taken very close to every cell in the body. White blood cells. White blood cells are easily recognized because unlike red blood cells they have a nucleus. The nucleus is often quite large. Many white blood cells can move around. They can squeeze out through the walls of blood capillaries into all parts of the body. Their function is to fight pathogens and to clear up any dead body cells. Some white blood cells destroy pathogens by engulfing them and digesting them in a process called phagocytosis. Figure 9.20. How a white blood cell destroys bacteria by phagocytosis. Other white blood cells produce molecules called antibodies. These molecules fix onto pathogens and help to destroy them. If you are studying the supplement, you will find out more about antibodies in Chapter 10. White blood cells that destroy pathogens by phagocytosis are called phagocytes. Most phagocytes have a lobed nucleus, like the ones in figures 9.19 and 9.20. White blood cells that produce antibodies are called lymphocytes. They usually have a large, round nucleus that almost fills the cell. Figure 9.21, electron micrograph of a lymphocyte 5000. Platelets. Platelets are small fragments of cells with no nucleus. They are made in the red bone marrow, and they are involved in blood clotting. Blood clotting stops pathogens getting into the body through breaks in the skin. Normally, your skin provides a very effective barrier against the entry of bacteria and viruses. Blood clotting also prevents too much blood loss. Blood plasma contains a soluble protein called fibrinogen. When a blood vessel is broken, the platelets release a substance that makes the fibrinogen change. The soluble fibrinogen becomes an insoluble protein called fibrin. This forms fibers, which pile up on top of each other to make a mesh-like structure that helps to seal the wound. Red blood cells get trapped in the mesh. The platelets stick together to form clumps. The fibers, the trapped red blood cells and the clumps of platelets form a blood clot. Figure 9.22, the sequence of events that causes a blood clot to form. Figure 9.23, this scanning electron micrograph shows red blood cells trapped in a tangle of fibrin fibers. Plasma. Blood plasma is mostly water. Many substances are dissolved in it. Soluble nutrients such as glucose, amino acids, and mineral ions are carried in the plasma. Plasma also transports hormones such as adrenaline and waste products, including carbon dioxide and urea. Table 9.2 summarizes some of the substances that are transported in blood plasma. Component. Source. Destination. Notes. Water. Absorbed in small intestine and colon. All cells. Excess is removed by the kidneys. Various proteins. Fibrinogen is made in the liver. Antibodies are made by lymphocytes. Remain in the blood. Fibrinogen helps in blood clotting. Antibodies kill invading pathogens. Table 9.2. Some of the main components of blood plasma. Activity 9.2. Summarizing the functions of blood components. Work in a group of 3 or 4 for this activity. You will need a large sheet of paper to make an illustrated table that can be displayed. In your group, discuss what your table will look like. You might like to have 3 columns. 1 for the component of blood, 1 for a description or picture of the structure of this component, and a third for a description of its functions. Try to make your table really easy to understand, colorful and eye-catching, full of relevant and clearly explained information. Peer assessment. When all the groups have completed their tables, they can all be displayed on the wall. Take some time to look carefully at the tables that other groups have produced. Make a note of at least one good point about each table, and at least one thing that could be improved. Share your ideas with the other groups, and listen to their ideas about your table. Is there anything that another group has done that you wish you had thought of? What do other groups like about your table? What do other groups suggest you could have done better? Questions. 21. State how you can distinguish a white blood cell from a red blood cell in a photomicrograph. 22. Write one sentence that explains, in your own words, what blood plasma is. 23. Outline 2 roles of blood clotting. 24. Explain how you can tell the difference between a phagocyte and a lymphocyte in a photomicrograph. 25. Blood plasma and platelets both play important parts in forming a blood clot. Describe how they do this. Summary. The mammalian circulatory system has blood vessels, a pump and valves. Fish have a single circulation, but mammals have a double circulation, where blood passes through the heart twice on one complete circuit. The heart has a muscular wall, a septum, left and right ventricles and atria, one-way valves and coronary arteries. The walls of the ventricles are thicker than those of the atria. The wall of the left ventricle is thicker than that of the right ventricle. The thicker walls contain more muscle, and can produce more force when it contracts. Atrioventricular and semilunar valves keep blood flowing in the correct direction. Blood is pumped away from the heart in arteries and returns to the heart in veins. Pulse rate, the sounds of valves closing, and ECGs can be used to monitor the activity of the heart. Coronary heart disease is caused when the coronary arteries become blocked. Risk factors for CHD include diet, sedentary lifestyle, stress, smoking, genes, age and gender. Arteries have a thicker wall than veins and a smaller lumen. Veins have valves. Capillaries have very thin walls. The differences between the structures of arteries and veins are related to the higher and more fluctuating pressure of blood in arteries than in veins. Each body organ is supplied with blood by an artery, and blood is taken away in a vein. Pulmonary vessels supply the lungs, and renal vessels supply the kidneys. Blood is made up of plasma, red blood cells, white blood cells and platelets. Red blood cells transport oxygen. White blood cells protect against pathogens by phagocytosis and producing antibodies. Platelets help with blood clotting. Plasma transports blood cells, ions, nutrients, urea, hormones and carbon dioxide. Phagocytes carry out phagocytosis, while lymphocytes produce antibodies. When blood clots, soluble fibrinogen is converted to insoluble fibrin, which forms a mesh and traps red blood cells. Project. Decreasing the risk of coronary heart disease. Work in a group of 3 or 4 for this activity. You are going to produce a leaflet that can help people to reduce their risk of developing coronary heart disease. Figure 9.24. A care worker talking a patient through a leaflet. Here are some things to think about as you design the leaflet. Who will read your leaflet? How will the leaflet be distributed to people? Keep it simple. People get confused if they are given too much information at once. Make it look attractive. A carefully thought-out picture or diagram can be much more interesting and easier to understand than a lot of words. Keep it positive. Do not make people feel hopeless about their present lifestyle. Try to help them to feel that they can easily make small changes that will make a difference. Be informative. People are much more likely to take action if they understand the reasons why it is important to do so. You can produce your leaflet by hand, or you can construct it using a computer or tablet. Exam-style questions. 1. What is a function of all arteries? ATO deliver oxygen close to every cell in the body. BTO ensure that blood flows in one direction. Only CTO pump blood around the body. DTO transport blood away from the heart. 2. Which component of blood transports dissolved carbon dioxide? A plasma. B. Platelets. Red blood cells. White blood cells. 3. The diagram shows a section through the heart. Which two areas contain deoxygenated blood? AP and QBQ, and RCR and S. DS and P. 4. The diagram shows some of the blood vessels supplying organs in the abdomen. What are blood vessels F, G and H? Vessel F. Vessel G. Vessel H. Aorta. Renal vein. Hepatic portal vein. Hepatic artery. Vena cava. Aortic vena cava. Renal artery. Hepatic vein. Adrenal vein. Hepatic vein. Vena cava. 6. The figure shows a white blood cell and a red blood cell. A. State two features of the white blood cell that differ from the red blood cell. B. The diameter of a red blood cell is 0.007 millimeters. Measure the diameter of the red blood cell in the figure. Use your measurement to calculate the magnification of the diagram. Show your working. Red blood cells transport oxygen. 5. What happens when blood clots? A. Insoluble fibrin is converted to fibrinogen. B. Insoluble fibrinogen is converted to fibrin. C. Soluble fibrin is converted to insoluble fibrinogen. D. Soluble fibrinogen is converted to insoluble fibrin. Explain how red blood cells take up oxygen, and how they transport it to respiring cells. D. Outline the function of white blood cells. 7. A. Outline two ways in which the activity of the heart can be monitored. B. Describe how the activity of the heart changes when a person exercises and then rests. C. The coronary arteries supply the muscle of the heart with oxygen. Describe the problems that occur if these arteries become blocked and discuss how a person can reduce the risk of this happening. 8. The graph below shows how the volume of the left ventricle changes over a time period of 1.2 seconds. A. Describe what the muscle in the wall of the left ventricle is doing at points A and B on the graph. Use the graph to determine the time for one complete heartbeat. C. Use your answer to B to calculate the heart rate in beats per minute. D. The atrioventricular valves close at point A. Explain how this helps the blood to circulate correctly. E. Make a copy of the graph. On your copy, sketch a line to show the volume of the right ventricle during this 1.2 second period. 9. The diagram shows a red blood cell inside a capillary. A. Structures A and B are inside a cell that forms the wall of the capillary. I. Identify structures A and B. 2. Explain why it is important that the wall of the capillary is only one cell thick. 3. Explain the advantages of the capillary being only just wide enough for the red blood cell to pass through it. B. Describe 3 ways in which the structure of the red blood cell differs from the cells in the wall of the capillary. C. Identify the liquid labeled C. Self-evaluation checklist. After studying this chapter, think about how confident you are with the different topics. This will help you to see any gaps in your knowledge and help you to learn more effectively. I can. C. Topic. Needs more work. Almost there. Confident to move on. Describe the structure of the circulatory system. 9.1. Describe the structure of the heart. 9.2. State that the activity of the heart can be monitored by measuring pulse rate, using ECGs, and listening to the valves closing. 9.2. Investigate and describe how exercise affects heart rate. 9.2. Describe coronary heart disease and list factors that increase the risk of developing CHD. 9.2. Describe the structures of arteries, veins, and capillaries. 9.3. Identify and name the main blood vessels to and from the heart, lungs, and kidneys. 9.3. Explain the advantages of a double circulatory system compared with a single circulatory system. 9.1. Explain the relative thickness of the walls of the atria and ventricles, and the left and right ventricles. 9.2. Describe the functioning of the heart in terms of the contraction of muscles and the action of valves. 9.2. Explain how exercise affects heart rate. 9.2. Chapter 10. Diseases and Immunity. In this chapter you will find out how pathogens are transmitted from one host to another. Learn how the body defends itself against pathogens. Explain what immunity is, and how it is produced naturally and through vaccination. Getting started. Here is a list of words that you have already used in your biology course. Antibody, bacterium, pathogen, virus, white blood cell. Try to write a sentence that includes all of those words. You can use plurals instead of singular words if you like. If you can't fit all the words into one sentence, try writing 2 sentences. Take turns with others to read out your sentence or sentences. Who has written the best sentence? What makes this the best sentence? Global outbreaks of measles. Measles used to be a very common disease, particularly of young children. Although many people who get measles recover fully, it can lead to serious complications and even death. In most countries, children are vaccinated against measles. The vaccine gives almost complete protection, and cases of measles fell dramatically when the vaccine first began to be used in the late 1960s and 1970s. However, in 2019 there was a significant increase in numbers of measles cases, with serious outbreaks happening in Ukraine, the Democratic Republic of the Congo, Madagascar, Angola, Cameroon, Chad, Kazakhstan, Nigeria, Philippines, South Sudan, Sudan, and Thailand. Numbers of cases also increased in developed countries such as the USA, which had its highest number of cases for 25 years. Worldwide, 3 times more cases of measles were reported in 2019 than in 2018. What happened? The virus that causes measles is very easily spread from one person to another. If at least 95% of people in a community are vaccinated against measles, then even the ones who are not vaccinated are unlikely to get the disease, because there are not enough hosts for the measles virus to live in. This helps to protect vulnerable children and adults, such as those who have a weak immune system. But not every country manages to vaccinate this percentage of children. And, increasingly, some parents are choosing not to have their children vaccinated. Figure 10.1. This boy has just been vaccinated against a number of common childhood diseases, including measles. Discussion questions. 1. Why might some parents choose not to have their children vaccinated against measles? 2. Some schools refuse to allow children who have not been vaccinated to come to classes. Do you think this is a good idea? 10.1 Transmission of pathogens. A pathogen is a microorganism that causes disease. Many diseases are caused by pathogens that get into our bodies and breed there. Table 10.1 shows the 4 kinds of microorganism that can act as pathogens, and some of the diseases that they cause. Figure 10.2, an electron micrograph of human immunodeficiency viruses. The colors are not real, they have been added to make the viruses show up more clearly. The blue circles are the protein coats, and the genetic material is contained in the red areas. Each virus is about 0.1 micrometers across. Group to which pathogen belongs. Examples of diseases which they cause. Viruses: influenza, common cold, poliomyelitis, measles, AIDS bacteria. Cholera, syphilis, whooping cough, tuberculosis, tetanus protoctists. Malaria, amoebic dysentery fungi. Athlete's foot, ringworm. Table 10.1, Types of pathogen. A person or other animal in which the pathogen lives and breeds is called a host. Diseases that are caused by pathogens can usually be passed from one host to another. They are called transmissible diseases. Once inside the body, some pathogens damage our cells by living in them and using up their resources. Others cause harm to cells and body systems by producing waste products, called toxins, which spread around the body and cause symptoms such as high temperature and rashes and make you feel ill. Some toxins produced by pathogens are among the most dangerous poisons in the world, for example, the one produced by the bacterium Clostridium botulinum. How pathogens enter the body. The movement of a pathogen from one host to another is called transmission. The entry of the pathogen into the body is known as infection. There are several ways in which transmission and infection can happen. Direct contact. Some pathogens pass from one person to another when there is direct contact between an infected person and an uninfected one. Diseases transmitted like this are known as transmissible diseases. For example, the virus that causes AIDS, called HIV, can be transmitted when an infected person's blood comes into contact with another person's blood. The fungus that causes the skin infection, athlete's foot, can be passed on by sharing a towel with an infected person. Indirect transmission. Most pathogens are transmitted indirectly. This can happen through breathing in droplets containing pathogens, touching a surface that someone with the pathogen has touched, eating food or drinking water that contains pathogens, contact with animals that are carrying pathogens. Cold, influenza and COVID-19 viruses are carried in the air in tiny droplets of moisture. Every time someone with these illnesses speaks, coughs or sneezes, millions of viruses are propelled into the air. If you breathe in the droplets, you may become infected. You can also pick up these viruses if you touch a surface on which they are present, and then put your hands to your face. Figure 10.3. A sneeze propels tiny droplets of moisture, containing millions of viruses, into the air. Catch your sneeze in a tissue. Bacteria such as Salmonella can enter your alimentary canal with the food that you eat. If you eat a large number of these bacteria, you may get food poisoning. Fresh foods, such as fruit and vegetables, should be washed in clean water before you eat them. Cooking usually destroys bacteria, so eating recently cooked food is generally safe. Food bought from street stalls is safe if it is hot and has just been cooked, but you need to take care with anything that has been kept warm for a while, as this gives any bacteria on it a chance to breed. Many governments make sure that food sellers are checked regularly to make sure that they are using good hygiene, and that their food is safe to eat. Figure 10.4. This food from a street stall is safe to eat because it has just been cooked, and the seller has reduced the risk of contaminating it with harmful bacteria by wearing gloves to protect the food. Many pathogens, including the virus that causes poliomyelitis and the bacterium that causes cholera, are transmitted in water. If you swim in water that contains these pathogens, or drink water containing them, you run the risk of catching these diseases. These pathogens can also get onto your hands if you touch anything that contains them, and then be passed into your body when you eat food that you have touched, or touch your mouth with your fingers. Figure 10.5. Contaminated water such as this almost certainly contains pathogens that would cause disease if they got into a person's body. Activity 10.1. Transmissible diseases. To start with, work on your own. Think about any transmissible diseases that you, or someone you know, may have had. Make a list. What kind of pathogen do you think caused each disease? How do you think that pathogen got into the body? Now share your ideas with your partner. Are there any transmissible diseases that you have both listed? Do you agree about the kinds of pathogen that caused these diseases? Do you agree about how the pathogen got into the body? If you do not agree, use the internet to check who is right. Reflection. When you are searching for reliable information on the internet, what tactics do you use? How do you decide on your search terms? How do you decide whether you can trust the information on a particular website? Body defenses. The human body has many natural defenses against pathogens. Some of them prevent pathogens from getting to parts of the body where they could breed. Figure 10.6 shows some of these defenses. Figure 10.6. Body defenses against the entry of pathogens. Pathogens that manage to get through all of these defenses are usually destroyed by white blood cells. In Chapter 9, we saw that some of these cells take in and digest the pathogens by phagocytosis, while others produce chemicals called antibodies that incapacitate or directly kill the pathogens. Questions. 1. Explain what is meant by a transmissible disease. 2. The pathogens that cause some transmissible diseases are transmitted by direct contact. How does the skin help to protect us against the entry of these pathogens? 3. How does stomach acid protect us against transmissible diseases? A clean water supply. We use water for many different purposes each day, especially for drinking, washing, and in preparing food. Water that has not been made safe to drink, for example, from a dirty container, or from a river or lake, can contain many different pathogens. Some of these are very dangerous, such as the bacterium that causes cholera, or the virus that causes polio. A clean water supply is one of the most basic health needs. In many countries, clean, safe water is supplied to homes and industries by a network of pipes. Before entering the pipes, the water is filtered to remove dirt, and then treated with chlorine to kill all the microorganisms in it. Where there is no piped water supply, people may be able to collect clean water from a communal source, such as a tap or deep well. Figure 10.7. Where clean water is not supplied through pipes, a deep well can be the best source of water. It is even better if the water is boiled before use, to kill any pathogens it might contain. Food hygiene. Good food hygiene makes it much less likely that someone eating the food you have prepared will get ill. Most food poisoning is caused by bacteria, so understanding the conditions that bacteria need for growth and reproduction can help us to keep them under control. A few simple rules can prevent you, or anyone else eating food you have prepared, from getting food poisoning. 1. Keep your own bacteria and viruses away from food. Always wash your hands before touching or eating food or putting your hands into your mouth for any reason. Keep your hair out of food. People working in food preparation environments often wear uniforms that cover their clothes and hair. Never cough or sneeze over food. Figure 10.8. Preparing food in a hospital kitchen. 2. Keep animals away from food. Animals are even more likely to have harmful bacteria on them than you are, so they should never be allowed to come into contact with food. Some are particularly dangerous. Houseflies usually have harmful bacteria on their feet because they may have been walking on rubbish, feces, or dead animals. And when flies feed, they spit saliva onto the food. Rats and mice often carry pathogens. Covering food to keep flies and other animals from touching it is always a good idea. Figure 10.9. Houseflies can transmit pathogens to food, on their feet and in their saliva. 3. Do not keep foods at room temperature for long periods. Figure 10.10 shows how bacterial growth and reproduction are affected by temperature. If there are even just a few harmful bacteria on food, these can reproduce and form large populations if the temperature is right for them. Keeping food in the fridge slows down bacterial growth. Cooking food at a high temperature will kill most bacteria. If cooked food is reheated, it should be made really hot, not just warmed. Figure 10.10, the effect of temperature on bacterial growth. 4. Keep raw meat away from other foods. Raw meat often contains bacteria. This is not a problem if the meat is to be cooked, because these bacteria will be killed. However, if the bacteria get onto other foods that might be eaten raw, then they might breed there. In any case, foods such as salads and vegetables that are to be eaten raw should be washed in clean water before eating, unless they have been packaged so that they cannot be contaminated with bacteria. Personal hygiene. Personal hygiene means keeping your body clean. This can greatly reduce the risk of getting, or passing on, transmissible diseases. We have already seen how important this is when preparing or eating food. Human skin makes an oil that helps to keep it supple and waterproof. If the skin is not washed regularly, this oil can build up, as can dirt from things that we have touched. When we are hot, we produce sweat from sweat glands in the skin. The evaporation of water from the sweat helps us to keep our body temperature from rising too high. If oil, dirt and sweat are left on the skin for long, they provide breeding grounds for bacteria. These can produce substances that smell unpleasant. Washing regularly, using soap and shampoo to help to remove oils, prevents this from happening. Figure 10.11. Washing with soap reduces the risk of transferring pathogens. There are also millions of bacteria inside our mouths. Most of these are harmless and may even be beneficial to us. But some of them can cause bad breath and tooth decay. Brushing teeth twice a day, and perhaps also using a mouthwash, can keep these harmful bacteria under control. Waste disposal. We produce an enormous amount of rubbish each year. Waste food, cardboard and paper packaging, bottles and cans, newspapers and magazines, plastic bags, old tires, anything that we have finished with and no longer want to use, are all thrown away. Sometimes, this waste simply accumulates close to where people live and work. Animals such as houseflies, rats and stray dogs may forage for food in the rubbish. Bacteria breed in the waste food. Dangerous chemicals seep out of the rubbish, polluting the ground and waterways. Figure 10.12. Waste, especially food waste left in the street, encourages rats and other animals, which can spread pathogens. In many countries, waste is regularly collected and taken to landfill sites. In some places, nothing is done to make the landfill site safe, but good management can avoid risk of infection and pollution. Figure 10.13 shows a well-designed landfill site. Only licensed operators are allowed to add material to the site, and the rubbish is checked as it is brought in, to make sure that nothing really dangerous is included. The rubbish is added in even layers, and is compacted to reduce the space it takes up. Figure 10.13, a well-constructed landfill site. Some of the rubbish in the landfill site is rotted by decomposers, especially bacteria. This produces a gas called methane, which is flammable, and could cause explosions if it is allowed to build up. Placing pipes in the rubbish can allow the methane to escape harmlessly into the air. Better still, the methane can be collected and used as a fuel. Eventually, when the landfill site is full, it can be covered over with soil and grass, and trees allowed to grow. Sewage treatment. Sewage is waste liquid that has come from houses, industry, and other parts of villages, towns, and cities. Some of it has just run off streets into drains when it rains. Some of it has come from toilets, bathrooms, and kitchens in people's houses and offices. Some of it has come from factories. Sewage is mostly water, but also contains many other substances. These include urine and feces, toilet paper, detergents, oil, and many other chemicals. Sewage should not be allowed to run into rivers or the sea before it has been treated. This is because it can harm people and the environment. Untreated sewage is called raw sewage. Figure 10.14. This open drainage canal contains rubbish and raw sewage, a major health risk to people living in this area. Raw sewage contains many bacteria and other microorganisms, some of which are likely to be pathogens. People who come into contact with raw sewage may get ill, especially if it gets into their mouths. Poliomyelitis and cholera are just two of the serious diseases that can be transmitted through water polluted with raw sewage. Activity 10.2. Controlling the spread of disease. Work in a group of 3 or 4 for this activity. Your task is to summarize the importance of a clean water supply, hygienic food preparation, personal hygiene, and safe disposal of sewage and other waste. Your summary should be in a form which can be shared with others. Decide on the form that your summary could take. It could be a series of slides, a video presentation, a poster, or an illustrated summary table. You can use the information in this book, or you may like to research more information on the internet. Peer assessment. Share your summary with another group or groups. Look at their summary and decide on at least 2 things that they have done very well. Then suggest 2 things that would improve their summary. Then consider the feedback that the other group gives to you. Do you agree with it? If you can, use their suggestions to improve your summary. Questions. 4. Explain why a clean water supply is very important to maintain health. 5. Suggest why a deep well can provide a safer source of water than a river or lake. 6. Explain why food that is not going to be eaten immediately should be kept in a fridge. Cholera. Cholera is a serious transmissible disease caused by a bacterium. Cholera bacteria can be spread through water and food that has been contaminated with feces from an infected person. In places where people are forced to live in unhygienic conditions, such as in refugee camps, cholera can spread very rapidly. A very serious cholera outbreak happened in Haiti in 2010, following a major earthquake that displaced thousands of people from their homes. At least 8,000 people were killed by this disease. Cholera bacteria live and breed in the small intestine, and produce a toxin that stimulates the cells lining the intestine to secrete chloride ions. These ions accumulate in the lumen of the small intestine. This increases the concentration of the fluid in the lumen, lowering its water potential. Once this water potential becomes lower than the water potential of the blood flowing through the vessels in the walls of the intestine, water moves out of the blood and into the lumen of the intestine by osmosis. Figure 10.15. This scanning electron micrograph shows a cholera bacterium. The long yellow-green thread is called a flagellum, and the bacterium uses this to move itself through a liquid. Figure 10.16. How the cholera toxin causes diarrhea. This is why cholera is so dangerous. The infected person suffers severe diarrhea, in which large quantities of water are lost from the body in watery feces. Without treatment, death may occur from dehydration and the loss of chloride ions from the blood. However, So long as enough fluids can be given to replace these losses, almost every person suffering from cholera will eventually recover. Questions. 7. Explain why cholera outbreaks are most likely to occur when people are displaced from their homes. 8. People with cholera can be successfully treated by giving them drinks containing water, glucose and salt. This is called rehydration therapy. Use the information in Figure 10.16 to suggest how these drinks can help them to survive. 10.2 The immune response. In Chapter 9, we saw that one type of white blood cell lymphocytes produces molecules called antibodies. Antibodies help to destroy pathogens. Antibodies. In your body, you have thousands of different kinds of lymphocyte. Each kind is able to produce a different sort of antibody. An antibody is a protein molecule with a particular shape. This shape is complementary to the shape of another molecule, which is called an antigen. Many antigens are molecules found on the outside of a pathogen. Each pathogen has its own antigens, which have specific shapes. To destroy a particular pathogen, antibody molecules must be made which are a perfect complementary shape to the antigens on the pathogen. The antibody molecules bind with the antigen. In some cases, this directly kills the pathogen. In other cases, the antigens stick the pathogens together. This stops the pathogens dividing or moving, making it easier for phagocytes to destroy them. Figure 10.17. This drawing shows antibodies binding to their antigens on a virus. The antibody molecules are the Y-shaped blue structures. Most of the time, most of your lymphocytes do not produce antibodies. It would be a waste of energy and materials if they did. Instead, each lymphocyte waits for a signal that a pathogen, which can be destroyed by its particular antibody, is in your body. If a pathogen enters the body, it is likely to meet a large number of lymphocytes. One of these may recognize the pathogen as being something that its antibody can destroy. This lymphocyte will start to divide rapidly by mitosis, making a clone of lymphocytes just like itself. These lymphocytes then secrete their antibody, destroying the pathogen. Figure 10.18. How lymphocytes respond to a pathogen. This takes time. It may take a while for the right lymphocyte to recognize the pathogen, and then a few days more for it to produce enough identical lymphocytes to make enough antibodies to kill the pathogen. In the meantime, the pathogen breeds, making you ill. Eventually, however, the lymphocytes, antibodies and phagocytes usually manage to destroy the pathogen, and you get better. Lymphocytes are a very important part of your immune system. The way in which they respond to pathogens, by producing antibodies, is called the immune response. Memory cells. When a lymphocyte makes copies of itself, not all of the new cells make antibodies. Some of them simply remain in the blood and other parts of the body, living for a very long time. They are called memory cells. If the same kind of pathogen gets into the body again, these memory cells will be ready and waiting for them. They will be able to make enough antibodies, very quickly, to kill the pathogens before they have time to produce a large population and do any harm. The person has become immune to that type of pathogen. Figure 10.19 shows how numbers of bacteria and antibodies in the body change after infection with a pathogen that your immune system has not met before, and when it infects you a second time. Figure 10.19, changes in the amount of antibodies and number of bacteria after a first and second infection. Questions. 9. Explain the difference between an antigen and an antibody. 10. A learner wrote that antibodies are the same shape as their specific antigens. What is wrong with that statement? 11. Look at the first graph in Figure 10.19. A infection happens at time 0. Explain why the number of antibodies does not begin to rise until some hours after this. B. Describe and explain what happens to the number of bacteria over the 4-day period. 12. Look at the second graph in Figure 10.19. Describe and explain what happens to the number of bacteria the second time a person comes into contact with the bacteria. 13. Predict and explain what would happen if the person is infected with a different kind of bacterium after an immune response like the one in Figure 10.19. Peer assessment. Exchange your answer to question 13 with a partner. Has your partner made a clear prediction about what would happen? Have they given a clear explanation of what they predict would happen? Give feedback to your partner about how well you think they have done this. Listen to their feedback to you, and make changes to your answer if necessary. Vaccination. In most countries, children are given vaccinations at various ages as they grow up. This is to immunize children against diseases caused by pathogens. Adults can also be given vaccinations if they are at risk of getting particular diseases. A vaccine may contain weakened or dead viruses or bacteria that normally cause disease. Some vaccines don't contain complete viruses or bacteria, but only their antigens. When the weakened pathogens or their antigens are introduced into the body, they are recognized by the lymphocytes that can make antibodies that have a complementary shape to their antigens. These lymphocytes multiply and produce antibodies just as they would after a real infection. They also make memory cells, which give long-term immunity. So, if the normal viruses or bacteria get into the body one day, they will be attacked and destroyed immediately. Some diseases require 2 vaccinations to provide really good, long-lasting immunity. After the first dose, the body makes enough antibodies and memory cells to provide partial protection against the pathogen. A second vaccination given a few weeks afterwards, sometimes called a booster, stimulates the lymphocytes and memory cells to make even more antibodies and even more memory cells. Active and passive immunity. A person has active immunity to a disease if their body has made its own antibodies and memory cells that protect against that disease. These memory cells can last in the body for many years. You can develop active immunity by having the disease and recovering from it. Being vaccinated. Active immunity can last a long time, sometimes an entire lifetime. The memory cells that were made during the infection or after the vaccination can spring into action at a moment's notice to make antibodies and stop an invading pathogen in its tracks. We can also be given a different kind of immunity, called passive immunity. A person has passive immunity to a disease if they have been given ready-made antibodies that have been made by another organism. Figure 10.20, Active and Passive Immunity. Babies get passive immunity by breastfeeding. Breast milk contains antibodies from the mother, which are passed on to her baby. This is useful because a young baby's immune system is not well developed, and so the mother's antibodies can protect it against any diseases to which she is immune for the first few months of its life. Another way of getting passive immunity is to be injected with antibodies that have been made by another organism. For example, if a person is bitten by an animal that might have rabies, they can be given antibodies against the rabies virus. These can destroy the virus immediately, whereas waiting for the body to make its own antibodies will take too long and the person is unlikely to recover. Active immunity is usually very long-lasting. Passive immunity, however, only lasts for a short time. This is because the antibodies will eventually break down. No lymphocytes have been stimulated to make clones of themselves. The body has not made memory cells, so any infection will be treated as a first-time one. Controlling disease by vaccination. Smallpox is a serious, often fatal, disease caused by a virus. It is transmitted by direct contact. If a person survives smallpox, they are often left with badly scarred skin and may be made blind. In 1956, the World Health Organization began a campaign to try to completely eradicate smallpox. They wanted to make the smallpox virus extinct. They set up systems to get as many people as possible, all over the world, vaccinated against smallpox. The campaign was a success. More than 80% of people in the world who were at risk from the disease were vaccinated. The very last case of smallpox happened in 1977, in Somalia. By 1980, 3 years had gone by with no more cases, and the WHO were able to declare that smallpox had been eradicated. Currently, attempts are being made to eradicate some other very serious diseases caused by viruses. One of these is poliomyelitis. Polio can infect children, and usually leaves them with permanent paralysis of parts of their body. Eradicating this virus is proving difficult, as people in some countries are resisting efforts to vaccinate children. Polio is now very rare in most parts of the world, but cases are still occurring in a small number of countries. Figure 10.21. A child being vaccinated for polio. The polio vaccine is unusual because it can be given by mouth. No injection is needed. The control of many other serious infectious diseases relies on vaccination of children. For example, we saw in the science in context at the start of this chapter that, in most countries, children are vaccinated against measles. Measles is spread by airborne droplets. It causes a skin rash and fever, and there can be very severe complications, such as blindness and brain damage. Vaccinating children against measles protects not only the children that are vaccinated, but also those that are not. This is sometimes called herd immunity. Vaccinating enough individuals protects the whole herd of people, even the ones who have not been vaccinated. Herd immunity works because there are fewer places for the measles viruses to replicate. They can only do so if they enter the body of an unvaccinated person. Activity 10.13. Vaccinations in your country. Work with a partner, or in a small group. Use the internet to find out what vaccinations are given to young children in your country. Do any of the vaccinations need to be given more than once, to provide good immunity? At what ages are the vaccinations given? Questions. 14. Outline 2 ways in which active immunity can be gained. 15. Outline 2 ways in which passive immunity can be gained. 16. Describe one difference between active immunity and passive immunity, other than the different ways that they are gained. 17. Explain how vaccination can help to control the spread of a transmissible disease. COVID-19. In December 2019, a new infection was detected. Scientists discovered that the pathogen that caused it was a new virus, which they named SARS-CoV-2. This stands for Severe Acute Respiratory Syndrome, Coronavirus 2. Coronaviruses are spherical viruses, with protein spikes sticking out. These spikes help the virus to attach to human cells. We already live with many different coronaviruses, which cause colds and flu. But this one is different. The illness it causes can often be very severe, and even fatal. Figure 10.22. This electron micrograph shows 4 SARS-CoV-2 coronavirus particles. The protein coat, with its spikes, has been artificially colored blue in this image 200,000. The disease caused by SARS-CoV-2 has become named COVID-19. You may like to try to work out what the letters and number in that name refer to. Learning about COVID-19. It took a little while for scientists to learn important facts about COVID-19. In particular, people needed to know how it is transmitted. This would help countries to plan how they could reduce the transmission. They also needed to find out how the virus affects the body, which would help to find ways to treat the disease. The virus is very easily transmitted from one person to another. It can be passed on in droplets, when a person coughs, sneezes or talks. It can remain on a surface, such as a door handle, for several days, and then get into another person's respiratory system if they touch the surface, and then their mouth. Because the virus spreads so easily, it quickly caused a pandemic, a disease that is present all over the world. The virus can even be transmitted from a person who does not have any symptoms. People can be completely unaware that they could be passing the virus on. In most people, the virus causes only mild symptoms. But in others, the lungs can be so badly affected that the person has great difficulty breathing. Even with excellent medical treatment, some of these people die. Scientists from many different countries worked together to discover all they could about the virus, how it is transmitted, what it does to the body, and how we could stop it. Medical staff across the globe focused their attention on finding ways to treat people who were suffering severe symptoms. Mathematical modelers used the information that different countries were providing about numbers of people infected with the virus, how many of them were ill enough to be admitted to hospital, how many of them died, and tried to predict what would happen next, and how different strategies might help to keep the numbers of infected and seriously ill people to a minimum. Many countries asked people to stay at home and not travel. Tests were developed to find out who had the virus, and some governments asked people who tested positive to isolate themselves for 14 days. In many countries, people who did go out were asked to wear masks. Figure 10.23. Research shows that wearing a mask not only reduces the risk of passing a virus to someone else, but also reduces the person's risk of inhaling it themselves. Activity 10.4. Researching COVID-19. COVID-19 is a new disease. You are going to research the current understanding of this disease and how people are affected by it. In your group, choose one of these topics to research. If you are not studying the supplement, then topic 1 is the best choice. Select websites carefully, ensuring that they are providing you with good scientific information. Use your findings to contribute to a display to put on the wall of the laboratory or your classroom. Topic 1. How did your country respond to the fight against COVID-19? What did your country do to try to stop this disease spreading when it first appeared? How does this compare with what other countries did? Why did different countries take different steps? Which worked best? How can we use this information to make better decisions if another pandemic arises in future? Topic 2. What does the virus do in the body? How does the virus infect cells? How does the immune system respond to infection with this virus? What have scientists learnt about why some people are more likely to become very ill than others? What treatments have proved to be most effective, and why? Topic 3. A vaccine against COVID-19. Producing and testing vaccines was done much more quickly than usual. How was this achieved? Have any vaccines proved to be successful? How do the vaccines work? Who is able to be vaccinated? How does this affect the lives of people in different countries of the world? Summary. Transmissible diseases are caused by pathogens, which can be passed from one host to another. Pathogens can be transmitted through direct contact with blood or other body fluids, or indirectly through contaminated surfaces, food, from animals, or through the air. The body has defenses to prevent the entry of pathogens, including skin, hairs in the nose, mucus, and stomach acid. White blood cells destroy pathogens that get through these defenses and enter the body. Each pathogen has its own antigens, which have specific shapes. Lymphocytes produce antibodies, which are proteins with specific shapes that are complementary to specific antigens. Antibodies bind with their antigens and either destroy the pathogen directly or make it easier for phagocytes to destroy the pathogen. Lymphocytes that have made contact with their antigen divide to form clones of memory cells. These remain in the body for long periods of time and can quickly respond to entry of pathogens with the same antigen at a later date. The production of antibodies and memory cells from the body's own lymphocytes leads to active immunity. This is long-lasting. Active immunity can result from having a disease and recovering from it or from vaccination. Immunity can also be gained by being given ready-made antibodies. Babies obtain antibodies from their mother when they feed on breast milk. Antibodies can also be given by injection. This is called passive immunity, and is not long-lasting because no memory cells are produced. Vaccination of a high percentage of a population helps to stop the spread of disease, because there are only a few unvaccinated hosts in which a pathogen can reproduce. Project: Finding a vaccine for malaria. Malaria is a serious disease that kills thousands of people each year, mostly children. The first malaria vaccine to be used on a widespread scale was launched in Kenya, Ghana, and Malawi in 2019. You are going to help to write a short play about the introduction of this vaccine, and then perhaps take part in the play. First, do some research about the first vaccine that was used in 2019. You could search on the following websites to begin with, but do look for others as well. World Health Organization website, and search for malaria vaccine. The History of Vaccines website, and search for malaria. The Zero Malaria Africa website. When you feel that you have collected enough information, begin to think about your play. It could involve characters like some of the following people: scientists trying to develop a malaria vaccine, or who have developed one that is now going to be used on a large scale for the first time. A philanthropist who is giving money to support the production and use of the malaria vaccine. Doctors who will be involved in the malaria vaccine campaign. Health workers who will be administering the vaccines. Parents who are wondering whether to let their child be given the vaccine. Children and/or adults who are going to be given the vaccine. Anyone else who might be involved in the project. Exam-style questions. 1. What is the correct term for a microorganism that causes disease? Antibody boasts pathogen. Vector. 2. Which disease is not transmissible? AIDS, caused by HIV. B. Coronary heart disease, caused by blockage in the coronary arteries. Influenza, caused by a virus. Malaria, caused by a single-celled organism. 3. What is a function of mucus in the passages leading to the lungs? A. Making it easy for air to move in and out of the lungs. B. Providing a low pH that kills pathogens. Secreting enzymes to kill bacteria. D. Trapping microorganisms before they reach the lungs. 4. Which statement about an antibody is correct? 8. Is a protein. B. It is found on the surface of a pathogen. CIT is secreted by phagocytes. D. It is the same shape as a specific antigen. 5. Which statements about active and passive immunity are correct? Active immunity. Passive immunity. A. Antibodies are secreted by lymphocytes. Antibodies are obtained from outside the body. B. Cannot be gained by vaccination. Can be gained by vaccination. C. Lasts for a short time only. Lasts for a long time. 6. A. Choose words from the list to complete the sentences about diseases. Bacteria, enzymes, host, mucus, pathogens, viruses. All transmissible diseases are caused by, which are passed from one to another. The lining of the respiratory passages helps to prevent this from happening by producing. B. Describe how the body defenses help to prevent bacteria entering, through the skin. II in food. B. The graph shows the number of cases of measles in the USA between 1944 and 2004. Measles is caused by a virus. Vaccination for measles was introduced in the USA in 1963. I. Describe how the number of cases of measles in the USA changed between 1945 and 1958. 2. Describe and explain the effect of the introduction of vaccination. 7. A. Explain how vaccination produces active immunity in a child. An aid worker is asked to travel immediately to an area where a disaster has taken place. There is a high risk of her being exposed to pathogens that cause serious diseases. Her doctor recommends that she should have an injection of antibodies, rather than a vaccination of weakened pathogens, before she travels. Suggest an explanation for the doctor's recommendation. C. Describe how antibodies help to destroy pathogens. 9. The electron micrograph shows a group of influenza viruses. 8. A. For each of the following events, decide whether it produces active immunity or passive immunity. Having a disease and recovering from it. Feeding a baby on breast milk. Being injected with antibodies. Being vaccinated with weakened viruses. A. Identify the part of the virus labeled A. B. New strains of the influenza virus frequently occur. The new strains have different proteins in their coats. Explain why a person who has recovered from influenza in 1 year may not be immune to influenza in the future. Self-evaluation checklist. After studying this chapter, think about how confident you are with the different topics. This will help you to see any gaps in your knowledge and help you to learn more effectively. I can. See topic. Needs more work. Almost there. Confident to move on. Explain what a pathogen is. 10.1. Describe how pathogens can be transmitted through direct contact, or indirectly. 10.1. Describe how the skin, hairs in the nose, mucus, stomach acid and white blood cells help to defend against pathogens. 10.1. Explain the importance of clean water, hygienic food preparation, personal hygiene, waste disposal and sewage treatment in controlling the spread of disease. 10.1. Explain what an antigen is, and state that each pathogen has its own antigens with a specific shape. 10.2. Describe antibodies as proteins secreted by lymphocytes, which bind to specific antigens with a complementary shape. 10.2. Outline how antibodies destroy pathogens, or help phagocytes to destroy them. 10.2. Explain what active immunity is, and how it is gained by an infection or vaccination. 10.2. Describe how memory cells are produced, and what they do. 10.2. Outline how vaccination can provide long-term immunity. 10.2. Explain how vaccination can help to control the spread of transmissible diseases. 10.2. Explain what passive immunity is, how it is gained and how it differs from active immunity. 10.2. Chapter 11. Respiration and gas exchange. In this chapter you will find out how the body uses energy, and how aerobic and anaerobic respiration supply this energy. Learn the equations for aerobic respiration and anaerobic respiration. Describe how gas exchange happens in the lungs, and the features of gas exchange surfaces. Investigate the differences in composition between inspired and expired air. Explain the differences in composition between inspired and expired air. Explain how breathing happens. Explain how activity affects breathing rate. Getting started. Sit quietly. Rest one hand against your sternum like the girl in the photo below. Think about the breathing movements you can feel. Now stand up and take 4 or 5 steps on the spot. Feel your breathing movements again. What happened? Why did this happen? Sleep apnea. Most of the time, we breathe regularly without even thinking about it. If you count your breathing movements, perhaps feeling the movements of your thorax as you breathe, you will probably find that you breathe in and out between 12 and 22 times each minute. Breathing moves air into and out of the lungs, providing oxygen that can be transported to all the cells in the body in the blood. Figure 11.1. You can feel your breathing movements by resting your hand just below the collarbone. Breathing is an automatic process, which happens even when we are asleep. But for some people, sleeping greatly disrupts their pattern of breathing. They seem to hold their breath for a long time, and then suddenly breathe in, sometimes with a snorting, gasping noise. People often wake themselves up when they do this. This condition is called sleep apnea. If it is severe, it can cause health problems. A person with severe sleep apnea can be helped by using a small machine to pump air into a mask that they wear over the mouth or nose while asleep. This helps to keep their breathing more regular, so that they can get a better night's sleep. Serious sleep apnea is more common in people who are very overweight, so losing weight can also help. It may also be helpful if they sleep on their side, rather than on their back. Stopping smoking is also a good idea. Discussion questions. 1. Breathing happens automatically, but we can also at least partly control it consciously. In contrast, we cannot control our heartbeat consciously. Can you suggest any reasons for these two processes being different in this respect? 2. Statistics show that people with untreated sleep apnea are more likely to have car accidents. Can you suggest why? 11.1 Respiration. Using energy. Every living cell needs energy. In humans, our cells need energy for contracting muscles, so that we can move parts of the body. Making protein molecules by linking together amino acids into long chains. Cell division, so that we can repair damaged tissues and can grow. Active transport, so that we can move substances across cell membranes up their concentration gradients. Growth, by building new cells which can then divide to form new cells. Transmitting nerve impulses, so that we can transfer information quickly from one part of the body to another. Producing heat inside the body, to keep the body temperature constant even if the environment is cold. All of this energy comes from the food that we eat. The food is digested, that is, broken down into smaller molecules, which are absorbed from the small intestine into the blood. The blood transports the nutrients to all the cells in the body. The cells take up the nutrients that they need. The main nutrient used to provide energy in cells is glucose. Glucose contains a lot of chemical energy. In order to make use of this energy, cells have to break down the glucose molecules and release the energy from them. They do this in a series of metabolic reactions called respiration. Like all metabolic reactions, respiration involves the action of enzymes. Aerobic respiration. Most of the time, our cells release energy from glucose by combining it with oxygen. This is called aerobic respiration. This happens in a series of small steps, each one controlled by enzymes. Most of the steps in aerobic respiration take place inside mitochondria. We can summarize the reactions of aerobic respiration as an equation: glucose plus oxygen, carbon dioxide plus water. The balanced equation is CH12O plus 6O6CO plus 6HO. Anaerobic respiration. It is possible to release energy from glucose without using oxygen. It is not such an efficient process as aerobic respiration, and not much energy is released per glucose molecule. This is called anaerobic respiration. Anaerobic respiration happens in the cytoplasm of a cell, not in mitochondria. Yeast, a single-celled fungus, often respires anaerobically. It breaks down glucose to alcohol. Glucose alcohol plus carbon dioxide CH12O2CHOH plus 2CO. You can see from the equation that carbon dioxide is made in this process. Plants can also respire anaerobically like this, but only for short periods of time. Some of the cells in your body, particularly muscle cells, can also respire anaerobically for a short time. But they do not do this in the same way as yeast. They make lactic acid instead of alcohol, and no carbon dioxide is produced. This happens when you do vigorous exercise, and your lungs and heart cannot supply oxygen to your muscles as quickly as they are using it. The muscle cells are able to release at least some energy from glucose without using oxygen, just to keep them going until oxygen is available again. Glucose lactic acid. Reflection. There are several equations to learn here, 3-word equations if you are studying the core only, plus 2 balanced equations if you are studying the supplement. You have already learnt the equation for photosynthesis in Chapter 6. How will you try to remember all of these equations, and not get them mixed up? Table 11.1 compares aerobic and anaerobic respiration. Table 11.1. A comparison of aerobic and anaerobic respiration. Questions. 1. Look at the equation for aerobic respiration. How does this compare with the equation for photosynthesis, which you learnt in Chapter 6? 2 inches. Which cells in your body does respiration happen? 3 inches. Respiration, energy is released from glucose. Think back to your earlier work, and explain how the energy became incorporated into the glucose molecules. Activity 11.1. Predicting the results of an experiment. Work with a partner for this activity. Look at experimental skills 11.1, investigating the effect of temperature on respiration in yeast. Using what you have learnt about respiration in this chapter, predict how temperature is likely to affect respiration in yeast. Hint. You may like to remind yourself of some of the information in Chapter 5. Sketch a graph to show your prediction. With your partner, write a sentence or 2 to justify your prediction. Experimental skills 11.1. Investigating the effect of temperature on respiration in yeast. In this investigation, you will measure the rate of respiration in yeast by counting how many bubbles of gas it releases in unit time. You will practice handling apparatus carefully, making measurements, recording results and displaying them in a suitable graph. You will need the apparatus shown in Figure 11.2. Retort stands, bosses and clamps to support it. A timer. Electronically controlled water baths, or make your own using beakers of water that you can heat or cool. A thermometer. Safety. Take care when using hot water. Water at 80 degrees Celsius is hot enough to burn skin. Figure 11.2. Apparatus for investigating respiration in yeast. Before you start, find out whether you will be using electronically controlled water baths or making your own. If you are going to make your own, make sure you know how you will do this. Whichever type of water bath you use, practice moving your apparatus into and out of a water bath without any of the tubes coming loose. Only the tube on the left in the diagram needs to be in the water bath. Method 1. Set up the apparatus shown in Figure 11.2. 2. Check the temperature of the electronically controlled water baths, or set up your own water bath. If you are making your own, decide on the temperatures you will use. If possible, use 5 different temperatures, perhaps 0 degrees Celsius as your lowest and 80 degrees Celsius as the highest, and 3 more in between. 3. Stand the tube with the yeast in it in the water bath at the first temperature. Leave the apparatus in the water bath for at least 3 or 4 minutes, to give time for the yeast and glucose mixture to come to the correct temperature. 4. Now start your timer, and count how many bubbles of gas are released into the liquid in the right-hand tube in 1 minute. Repeat twice more at this temperature. 5. Repeat steps 3 and 4 at the 4 other temperatures you have chosen. 6. Record all of your results in a suitable chart. Remember to calculate the mean number of bubbles produced at each temperature, and include that in your results chart. 7. Plot a line graph with temperature on the x-axis, and mean number of bubbles per minute on the y-axis. Questions. 1. Did you have any anomalous results, that is, results that did not fit the general pattern? If so, what did you do about them? 2. Describe the shape of your graph. 3. Use your knowledge of enzymes to explain the shape of your graph. 4. How well did the results of your experiment match the prediction that you made in Activity 11.1? If they were not the same, can you suggest why? 5. Explain why we cannot tell if the yeast was respiring aerobically or anaerobically during this experiment. 6. If the experiment is continued for a long time, no more bubbles are produced. Suggest why this happens. Self-assessment. How well did you manage this experiment? Rate yourself for each of the statements using: if you did it really well, if you made a good attempt at it and partly succeeded, if you did not try to do it, or did not succeed. I used at least 5 different temperatures. I managed the water baths well. I moved the apparatus into and out of the water baths without letting anything fall apart. I counted the bubbles accurately. I kept myself and others safe while doing the experiment. 11.2 Gas exchange in humans. Gas exchange surfaces. If you look back at the aerobic respiration equation near the beginning of this chapter, you will see that 2 substances are needed. They are glucose and oxygen. The way in which cells obtain glucose is described in chapters 6 and 7. Animals get sugars such as glucose from carbohydrates which they eat. Plants make theirs by photosynthesis. Oxygen is obtained in a different way. Animals and plants get their oxygen directly from their surroundings, either from the air for terrestrial organisms, or from oxygen dissolved in water for aquatic ones. If you look again at the aerobic respiration equation, you can see that carbon dioxide is made. This is a waste product and it must be removed from the organism. In organisms, there are special areas where the oxygen enters and carbon dioxide leaves. One gas is entering, and the other leaving, so these are surfaces for gas exchange. The gas exchange surfaces have to be permeable, so that oxygen and carbon dioxide can move easily through them. They have other characteristics which help the process to be quick and efficient. They are thin to allow gases to diffuse across them quickly. They are close to an efficient transport system to take gases to and from the exchange surface. They have a large surface area, so that a lot of gas can diffuse across at the same time. They have a good supply of oxygen. The human breathing system. Figure 11.3 shows the structures which are involved in gas exchange in a human. The most obvious are the 2 lungs. Each lung is filled with many tiny air spaces called air sacs or alveoli. It is here that oxygen diffuses into the blood, so the surface of the alveoli is the gas exchange surface. Because they are so full of air spaces, lungs feel very light and spongy to touch. The lungs are supplied with air through the windpipe or trachea. Figure 11.3, the human gas exchange system. The nose and mouth. Air can enter the body through either the nose or mouth. Hairs in the nose trap dust particles in the air. Inside the nose are some thin bones, which are covered with a thin layer of cells. Some of these cells, called goblet cells, make a liquid containing water and mucus. The water in this liquid evaporates into the air in the nose and moistens it. Figure 11.4, part of the lining of the respiratory passages. Other cells have very tiny hair-like projections called cilia. The cilia are always moving, and bacteria or particles of dust get trapped in them and in the mucus. Cilia are found all along the trachea and bronchi, too. They sweep the mucus, containing bacteria and dust particles, up to the back of the throat, so that it does not block the lungs. The trachea. From the nose or mouth, the air then passes into the windpipe or trachea. Just below the epiglottis is the voice box or larynx. This contains the vocal cords. The vocal cords can be tightened by muscles so that they make sounds when air passes over them. The trachea has rings of cartilage around it. As you breathe in and out, the pressure of the air in the trachea increases and decreases. The cartilage helps to prevent the trachea collapsing at times when the air pressure inside is lower than the pressure of the air outside it. The bronchi. The trachea goes down through the neck and into the thorax. The thorax is the upper part of your body from the neck down to the bottom of the ribs and diaphragm. In the thorax, the trachea divides into two. The two branches are called the right and left bronchi. One bronchus goes to each lung, and then branches out into smaller tubes called bronchioles. Alveoli. There are many tiny air sacs or alveoli at the end of each bronchiole. This is where gas exchange takes place. Figure 11.5, alveoli in the lungs, and the blood vessels that are associated with them. As we have seen, the walls of the alveoli are the gas exchange surface. Tiny capillaries are closely wrapped around the outside of the alveoli. Oxygen diffuses across the walls of the alveoli into the blood. Carbon dioxide diffuses the other way. Figure 11.6, gas exchange across the wall of an alveolus. The walls of the alveoli have several features, which make them an efficient gas exchange surface. They are very thin. They are only 1 cell thick. The capillary walls are also only 1 cell thick. An oxygen molecule only has to diffuse across this small thickness to get into the blood. They have an excellent transport system. Blood is constantly pumped to the lungs along the pulmonary artery. This branches into thousands of capillaries, which take blood to all parts of the lungs. Carbon dioxide in the blood can diffuse out into the air spaces in the alveoli, and oxygen can diffuse into the blood. The blood is then taken back to the heart in the pulmonary vein, ready to be pumped to the rest of the body. They have a large surface area. In fact, the surface area is enormous. The total surface area of all the alveoli in your lungs is over 100 m. They have a good supply of oxygen. Your breathing movements keep your lungs well supplied with oxygen. This is called ventilation. Questions. 4. List, in order, all of the structures that a molecule of oxygen passes through as it moves from the air, down into the lungs, and into the blood. 5 inches. Figure 11.6. Look carefully at the arrows showing the movement of oxygen into the blood, and carbon dioxide out of the blood. Apart from going in different directions, what other difference can you see between them? Explain this difference. 6. Look at figure 11.6 again. Some of the movement indicated by the arrows is mass flow, where a lot of air all moves in the same direction. Some of the movement is by diffusion, where individual molecules move randomly. Which arrows represent which type of movement? 7. People who smoke cigarettes often develop an illness called emphysema, where the walls of the alveoli break down. Instead of having millions of tiny air spaces in their lungs, they have fewer larger ones. Predict how this might affect the person's ability to do exercise. Explain your prediction. Experimental skills 11.2. Investigating the difference in composition between inspired and expired air. In this investigation, you will use lime water to compare how much carbon dioxide there is in the air you breathe in and the air you breathe out. You will practice observing carefully, and then use your observations to draw a conclusion. You will need the apparatus shown in figure 11.7. Figure 11.7, apparatus for comparing the carbon dioxide concentration in inspired and expired air. Safety, lime water is an irritant. Do not get it in your mouth. Wear safety goggles when breathing in and out through the tubing. Make sure that you do not suck hard through the rubber tubing. Take care that the rubber tubing is really clean before you put your mouth over it. Use a new piece of tubing for each person who uses the apparatus. Method 1. Look carefully at the apparatus, especially at the lengths of the glass tubes. When someone breathes gently into the rubber tubing, in which test tube do you predict bubbles will appear? 2. Breathe gently into and out of the rubber tubing. Take care not to suck too hard. You want to make bubbles appear in the lime water, not suck lime water into your mouth. Keep doing this until the lime water in one of the test tubes becomes cloudy. 3. Continue breathing in and out for a while. You may find that the lime water in the other test tube also becomes cloudy. Questions. 1. Explain why bubbles appeared in different test tubes when you breathed in and when you breathed out. 2. Explain why the lime water went cloudy more quickly in one of the test tubes. Comparing inspired air and expired air. Table 11.2 summarizes the differences between the composition of inspired air and expired air. Component. Percentage in inspired air. Percentage in expired air. Oxygen 21. 16. Carbon dioxide 0.04. For water vapor. Variable. Usually very high. Table 11.2. A comparison of inspired air and expired air. The air that we breathe in usually has an oxygen content of about 20 to 21%. There is less oxygen in the air that we breathe out, because cells in the body use oxygen in respiration. However, expired air does still contain a significant amount of oxygen, usually about 16%. The main reason for this is that our expired air mixes with normal air in the bronchi and trachea, so that what we actually breathe out is a mixture of the air in the alveoli and atmospheric air. The air we breathe in contains only a very small percentage of carbon dioxide, usually about 0.04%. Body cells produce carbon dioxide in aerobic respiration, and this carbon dioxide diffuses out of the blood and into the alveoli. Extra carbon dioxide is therefore present in the air that we breathe out, increasing its concentration to about 4%. Breathing movements. To make air move in and out of the lungs, you must change the volume of your thorax. First, you make it large so that air is sucked in. Then you make it smaller again so that air is squeezed out. This is called breathing. Muscles in two parts of the body help you to breathe. Some of them, called the intercostal muscles, are between the ribs. The others are in the diaphragm. The diaphragm is a large sheet of muscle, an elastic tissue which stretches across your body, underneath the lungs and heart. Figure 11.8. The rib cage and intercostal muscles. Breathing in. When breathing in, the muscles of the diaphragm contract. This pulls the diaphragm downwards, which increases the volume in the thorax. At the same time, the external intercostal muscles contract. This pulls the rib cage upwards and outwards. This also increases the volume of the thorax. Figure 11.9. How breathing movements change the volume of the thorax. As the volume of the thorax increases, the pressure inside it falls below atmospheric pressure. Air therefore flows in along the trachea and bronchi into the lungs. Breathing out. When breathing out, the muscles of the diaphragm relax. The diaphragm springs back up into its domed shape because it is made of elastic tissue. This decreases the volume in the thorax. The external intercostal muscles also relax. The rib cage drops down again into its normal position. This also decreases the volume of the thorax. Usually, relaxing the external intercostal muscles and the muscles of the diaphragm is all that is needed for breathing out. Sometimes, though, you breathe out more forcefully when coughing, for example. Then the internal intercostal muscles contract strongly, making the rib cage drop down even further. The muscles of the abdomen wall also contract, helping to squeeze extra air out of the thorax. Questions. 8. Suggest why the quantity of water vapor in expired air is always high. 9. Nitrogen is soluble in water, and molecules of nitrogen gas can pass easily through cell membranes. Body cells cannot use nitrogen gas. Inspired air contains about 78% nitrogen. Predict the percentage of nitrogen in expired air and explain your prediction. 10. Construct a table to show what the diaphragm, external intercostal and internal intercostal muscles do when you are breathing in, and when you are breathing out. Activity 11.2. Explaining the meanings of terms. Many people use the words breathing, respiration, and gas exchange as though they mean the same thing. Work in a small group to discuss the meaning of these three terms. Try to write a short, clear description of each one, which makes clear the differences between them. Share your ideas with other groups. Use the best ideas from all the groups in your class to write a really clear description that you think will help everyone to use the words correctly. Experimental Skills 11.3. Investigating the effect of physical activity on rate and depth of breathing. In this experiment, you will investigate how exercise affects your breathing. You will plan your own experiment, using the experience you gained when you did Experimental Skills 9.1, investigating the effect of physical activity on heart rate. This investigation gives you practice in planning an experiment where you change one variable, measure another, and try to keep other important variables the same. You will need a timer. Safety. If you are worried that doing exercise might make you ill, do not do it. You can measure someone else's breathing rate instead. Before you start, practice measuring breathing rate and depth. Work with a partner, and decide how you will count how many breaths you take per minute. For breathing depth, you may need to just observe any changes, rather than trying to measure them. Make a firm decision about how you will do this. Method 1. Make a plan of what you will do. Think about how you will change your independent variable. You may find it helpful to look at the instructions for Experimental Skills 9.1, investigating the effect of physical activity on heart rate. Or, think about how you did that investigation. 2. Construct a results table. 3. Do your experiment and record your results in your table. Be prepared to make changes to your original plan if you realize that this would improve your experiment. Questions. 1. Use your results to draw a line graph. Put time minutes on the x-axis and breathing rate breaths per minute on the y-axis. Mark on the x-axis where exercise began and ended. Use a ruler to join the points with straight lines. 2. Use your table or graph to describe, in words, how exercise affected breathing rate. 3. Evaluate the reliability of your results. Do you think you would get the same results if you did your experiment again tomorrow? Explain why you think this. Exercise and breathing rate. All the cells in your body need oxygen for respiration, and all of this oxygen is supplied by the lungs. The oxygen is carried by the blood to every part of the body. Sometimes, cells may need a lot of oxygen very quickly. Imagine you are running in a race. The muscles in your legs contract much harder and more frequently than usual, so they require more energy. The cells in the muscles combine oxygen with glucose as fast as they can to release extra energy for muscle contraction. A lot of oxygen is needed to work as hard as this. You breathe deeper and faster to get more oxygen into your blood. Your heart beats faster to get the oxygen to the leg muscles as quickly as possible. Eventually a limit is reached. The heart and lungs cannot supply oxygen to the muscles any faster. But the muscles continue to contract quickly, requiring more energy. How can that extra energy be found? Extra energy can be provided by anaerobic respiration. The muscle cells continue to respire aerobically with the oxygen they have, but they also break down some glucose without combining it with oxygen. Glucose lactic acid plus energy. As you saw in topic 11.1, anaerobic respiration does not release very much energy, but a little extra might make all the difference. When you stop running, you will have quite a lot of lactic acid in your muscles and your blood. This lactic acid must be broken down by combining it with oxygen in the liver. This is done by aerobic respiration in the liver cells. So, even though you no longer need extra energy, you continue to breathe faster and more deeply than normal, and your heart rate continues to be high. You are taking in and transporting extra oxygen to break down the lactic acid. The faster heart rate also helps to transport lactic acid as quickly as possible from the muscles to the liver. While you were running, you built up an oxygen debt. You borrowed some extra energy, without paying for it with oxygen. Now, as the lactic acid is combined with oxygen, you are paying off the debt. Not until all the lactic acid has been used up, do your breathing rate and heart rate